Okay.
Just as unbelievable to me, too.
Uttam RajBhandary, known to most of us as merely as Tom. Tom grew up in Nepal, spent his early years there, in his elementary education in Nepal. And he got his early collegiate education in Nepal as well, a college that was associated in Nepal with the University of Patna in India. So he received his bachelor's degree in 1952 from the University of Patna, and then enrolled in the master's program at the University of Calcutta in India, and received the Master of Science degree there in 1955. He then immigrated to England and began his PhD work at King's College at the University of Durham under the mentorship of Jim Baddeley. I knew Baddeley quite well at that time because of our mutual interest in coenzyme A, although I didn't know Tom at that time. Thomas already, when he introduced me, said that he knew of me at that time at least. He got his PhD in 1962, and then immediately came to this country to work with Goban Khurana, who at that time was at the Enzyme Institute at the University of Wisconsin. So Tom joined Goban at the University of Wisconsin, and 2 years later, in 1964, Tom was made a faculty member at the University of Wisconsin. In the late 1960s, we here at MIT were trying very hard to recruit Goban Khurana to come here to MIT. And we were able finally to get him to agree to come here. But as a bonus, we also convinced Tom to come along as well. And that was a real bonus for us. So Tom arrived here in 1969 and became a faculty member here in the biology department. In 1969, Vernon Ingram and I had been teaching general biochemistry together for about 10 years. And shortly after Tom arrived, he took Vernon's place in this course and took over the teaching of nucleic acid biochemistry and protein structure. This are areas that Vernon had been teaching. I had been teaching enzymology and metabolism in the general course, 705, as you all remember. And so he took over for Vernon at that time. And for the next 20 years or so, Tom and I taught that course together, 705. And of course, we got to know each other very, very well during that period of time. Not only were we teaching colleagues, but it turns out that somewhere along the line, because of some interest that in my own laboratory having to do with how the enzymatic methylation of nucleic acid takes place, Tom and I got entangled in research as well. So we not only became close colleagues, teaching colleagues, but we became at that time research colleagues as well. Well, not only did we become good research and teaching colleagues, but we also became very good personal friends. And that friendship has extended right up until the present time. And it is a real pleasure this morning for me to introduce Uttam RajBhandary to you, where he will tell you about his, whatever he's asked to tell you. Be sure and ask him, be sure somebody asks him about his early years in Nepal. Okay, Tom, the floor is yours.
Thank you, Gene. Thank you for your kind words. Thank you. Yeah. Gene, as always, is very kind, and I think I have one of the things that I've really enjoyed about being at MIT is the opportunity to have colleagues like Gene and everybody here, and also to collaborate with people over the years. So that's been really wonderful. So what can I tell you about my early years in Nepal? Manda wanted me to say a few words, so let me just digress here. I grew up in a family with 9 brothers and sisters. I was the oldest son, and in Nepal, well, I grew up in the valley of Kathmandu, and the valley is approximately 10 miles by 12 miles, and at that time had a population of about 150,000, I think. It's swollen immensely now. It had only one college. All of Nepal did not have a university. It had one college in Kathmandu, which is 2 miles from my house, and that's where I got my education, college education. But before that, I also went to school locally. And I have to confess to something here. I really spent only 5 years at school. Much of my education was really at home through tutors. My father wanted me to have a good background, so he hired people to teach me English and geography and Sanskrit and all the stuff. But mostly, many of them were actually not hired so much. They were actually my cousins. Cousins who really were good geographers, cousins who really knew mathematics and things like that. So that was really wonderful to learn from them. I spent only 5 years at school. And then I went to college. Now, many of you here, I think, you at the age of 10 or 12, you knew exactly what you wanted to be. That was not the case with me. I had no clue as to what I wanted to be. But the expectation was that I was the oldest son, and that you would go into a profession that would earn money, and help the family, right? So, and the only profession one could think of is medical profession, be a doctor and be famous, earn money. So I went to college, and in the system is that to get a bachelor's degree, you get 2 years to get what's called an intermediate in science, and then 2 years after that to get bachelor's. And when I finished my intermediate in science, I found out that I had neglected to take botany and zoology, that it was necessary for you to get into medical school. So either I had to stay 1 more year and take botany and zoology, or move on. And what did I do? I moved on. I moved on and decided to just get a bachelor's degree in science with physics, chemistry, and mathematics, which is what I did. And I was fortunate to do well enough that at that time, some of the older colonies, India, Pakistan, Ceylon, all those things, the Britain had pulled back. And so they formed a union they actually had a plan to help each other. They still were part of the British Commonwealth. They formed a plan to help each other through something called a Colombo Plan. The Colombo Plan because, they met in Colombo, in Ceylon, at that time Ceylon, now Sri Lanka. And so they decided to help each other through establishing scholarships and things like that. And although Nepal was never a part of the British Commonwealth because it was not colonized by Britain, they somehow felt that Nepal was a needy country and that they would want to help us. So there were 2 or 3 scholarships to people from Nepal, and I was fortunate enough to get one of those to go to Calcutta to do my master's in chemistry. And there, unfortunately, I fell ill, so I lost a year, and then I went back again in '53, and just basically finished my degree in chemistry, taking physical, inorganic, and organic chemistry. So I'm a chemist throughout, But then, fortunately, again, I did well enough there that I got a scholarship to go to Britain, the Colombo Plan scholarship again. And I had no say as to where I went. Just the people decided for me as to where I should go. All they want to know was what do you want to do. I said I majored in organic chemistry, so I would do organic chemistry. And then I was sent to Jim Baddeley's lab, and Jim had done his work. He's a chemist, had done his work with Alexander Todd, who was a professor of chemistry at Manchester and then at Cambridge. And Todd, as some of you might know, got the Nobel Prize, became well known. He was in the House of Lords and et cetera, in England. So Baddeley had done his work with Todd, a postdoc somewhere in Sweden. Then spent a year in Lippmann's lab, which is, I think, where he got interested in coenzyme A, that Gene was talking about, and spent some years at the Lister Institute in London. And then he got a professorship in Newcastle, and that's where I was sent, to Newcastle. Now, the time I went to Newcastle was, it was kind of time of much international tension. It was kind of almost, if you were superstitious, you might say it was ominous, because Britain and France had just invaded the Suez Canal. The Russians were threatening to bomb Britain at that time. This was 1956. And I was on my way to Britain. So off I went anyway. I remember vividly the airports that we had. No, there's no direct connection between Kathmandu and London, of course. And we flew in an old DC-3, the only planes that could fly from Kathmandu to Delhi. And then from Delhi, I remember all the stops in Karachi, Pakistan, Tehran, Basra in Iraq, Istanbul in Turkey, Frankfurt in Germany, and then London. Or the British Overseas. No, it's British Overseas Airlines Corporation. So BOAC at that time. This was 1956. Got it to Newcastle and started working with Baddeley. And it was my thesis project was synthesis of coenzyme A. And that's really where I had to read a lot of literature on coenzyme A. I came across Gene's early work with Esmond Snell on pantothenic acid, pantothane, et cetera, et cetera. So, and there, I think, learned synthetic chemistry and peptide chemistry, sulfur chemistry, nucleotide chemistry, and things like that, which I think really was the beginning of my scientific career, I have to say. That really was a steep learning curve for me. But but then at the end of 3 years, when what happened was, before I finished my project, which is synthesis of coenzyme A, someone else synthesized it. And, and that someone else is Gobind. Right? So Gobind had a student named John Moffat, and John synthesized coenzyme A. So my thesis title became Studies in the Synthesis of Pyrophosphates Related to Coenzyme A. So anyway, but then I had the choice. Baddeley said, okay, so you can do one of two things. Either you can write a thesis, and then you can, he also thought that I should stay there as a postdoctoral fellow. And I had already interviewed for an ICI fellowship and an MRC fellowship, and I got the MRC fellowship. And so I had a choice of either writing my thesis and then staying on for the fellowship, or I wanted to go home for a while. So I went home, back to Nepal for 2 months. And when I came back, instead of writing my thesis, Baddeley said, well, we're running into some competition here. On another project that he was working on, Baddeley had discovered molecules called teichoic acids. And teichoic acids are these polymers in bacterial cell walls. Gram-positive bacteria have these cell walls, polymers. And he was running into competition from Jack Strominger here. And so I said, okay, so I'll do that. And so I did that, worked on that project for 3 years. Once again, learned a lot about, phosphate chemistry, amino acid chemistry. But although it was all chemical work, all the analytical techniques that we used were all really biochemical. This chromatography, electrophoresis, colorimetric analyses, and things like that. So that really got me interested more in biochemistry at that time, and moved from chemistry to biochemistry. And then when Baddeley got a letter from Gobind, asking for people from his lab who might want to do postdoctoral work. And Baddeley came and asked me, well, are you interested in working with Gobind? Well, I said, yeah, of course. So then that's what brought me to Wisconsin. And again, that time was another ominous time, because that was a time of Cuban Missile Crisis. And again, the Russians threatening America with all kinds of stuff, and I was on my way to America. So I remember again, took the boat, Queen Mary, from Southampton to New York. Left my wife and a 6-month-old child because I wanted to get settled and have an apartment before they came over. And I'd never really experienced seasickness in my life, and did. And arrived in New York. The dockers were on strike. I had no clue how to do. When dockers are on strike, you have to handle your own luggage and things like that. And I came with quite some luggage. But anyway, so it was a struggle. Stayed at the YMCA for one night. Shipped all the stuff to Madison. Somebody helped me ship all the stuff to Madison. Then the next day, I took the train to Madison. And when I got to Gobin's lab, I was absolutely thrilled to know that Gobin wanted me to work on transfer RNAs, sRNA at that time, called transfer RNA, because I thought that I really was very interested in generally in questions of structure and function. And here was a class of molecules that had been discovered just a few years ago, and which obviously played a very important role in protein synthesis. So that's really what led me to work on transfer RNAs, and I have stayed with that and translation ever since. So I think, and at Madison was a very, very exciting place to be. Because at that time, again, this is 62, was the heydays of molecular biology, right? There were some very young people there who'd just been hired, people like Hatch Eccles, Bill Dove. These are really, really well-known guys in the field of lambda genetics and biology. Julius Adler was there. Masayasu Nomura had just been hired. Chuck Curlin had just been hired, and really a wonderful place to be, and where I learned a lot from just being with these people, and learned to grow 150 liters of lambda infected cells, and actually, isolating lambda phage, centrifuges, day and night centrifuges. You can imagine banding, cesium chloride banding of phages from 150 liters of culture. Yeah. Well, I don't. Yeah. So it was a good experience. It was, and then, as Gene said, I got into faculty. I was appointed a faculty member there. Didn't have to teach much. I just have to give 4 lectures a year, and this was a multi-team taught course. Again, molecular biology oriented, but my lectures were mostly chemical, nucleic acid chemistry and synthesis types of lectures. So I enjoyed my time immensely there. 7 years. Very difficult place to leave because it's a lovely, lovely, lovely city, great university, lakes all over, and children were happy. But then, when MIT came, there was no choice. No choice but to come here, and I think I have really enjoyed being here ever since. 1969 is when we came, and it's been 38 plus years, year 39 now. And over the years, wonderful colleagues, you see many of them here, and just been fabulous. I've collaborated with, my first collaboration was with Harvey Lodish. And this was work done by David Hausman. David Hausman was at that time a student enrolled at Brandeis, but working in Harvey's lab. And so David and I collaborated, and we published a paper. The very first paper I published from MIT was with Harvey. And then we collaborated with Gene. Gene had just come back from Caltech, I think, working with Attardi on a sabbatical, and working with RNA methylases. And so we used those enzymes to introduce methylations into tRNAs and look to see what the effect was on structure and function and things like that. Collaborated with Govind over the years, Alex, Phil Sharp, and several others, really. And so it's been a wonderful time of collaboration, camaraderie, teaching, and I think taught with Gene. Then went on to teach molecular biology first with Barbara for a year, and then with Alan Grossman. Then taught introductory biology with one of many people, David Hausman, David Page, Eric Lander, and Now, then taught introductory lab course 702. And now I have the privilege of teaching 725, a new course, completely new course right now, so with Boris Magasan. So that's where things stand, and I will answer questions to the best of my ability here.
So I have asked him to stop for 30 minutes, but he's calling his boss to see if he can stop.
Okay. But you can hear me, right? I'll try to yell. Okay, yeah.
Oh, nobody can hear you in the back.
Nobody. I'm sorry. Okay.
So will it be all?
Numerous siblings end up going to the West also. Can you repeat the question?
Okay. Did any of my numerous siblings end up going west? The only one who did was, for a few years, was my sister. My sister went to Germany to study archaeology. And so that was, I think, again, through scholarships that the Germans provided. And she went back to Nepal, and she just...
What did your father?
My father, yeah. Well, my father was a civil servant. He was in the customs department. He eventually rose to be the director of customs, and was that for many, many years. But it's really, I have to kind of say that my father was my guiding light, really, because I think from him I learned, if you are a chief of customs in Nepal, you can make a lot of money. You can make a lot of money, and yet we did not. He was just in terms of honesty and integrity and all that, really, and keeping one's word. So he was my guiding light. And he was a civil servant, lowly at the time I was a student, but then he eventually rose up to be the chief of customs.
So how did your family take it when you didn't become a doctor?
I think they did. I think they took it okay because I was doing something else. But I think what really shocked me was the fact that I did not go back to Nepal. Because actually, I have to tell you that when I moved from England to here, we shipped everything we owned to Nepal, with the idea that we would be in this country for 2 years. And 2 years became 3 years and 4 years, and after 4 years, and you really get into research and all that. There's no way you can go back to Nepal because that was I think you get bitten by the research bug and scientific interests and things like that. And,
what was the expectation in your family for girls going to college? I know that the sister was the archaeologist.
I think most of my sisters, particularly younger ones, ended up going to college in Nepal. Yeah. So by the time I left, I think there were several colleges had been established, women's college, and so there was the expectation, at least among in certain social circles, that the children would go to college. Yeah. So there was this kind of a expectation and emphasis on learning, and that's, yeah. So one of my other sisters teaches or taught at the university for several years. The sister who went to Germany, she ended up being director of the archaeology department and then became the secretary. And many of our colleagues who have been to Nepal know her because she takes them everywhere. And wherever she goes to, to some archaeological place, some palace or another, all that, and I think, she can take them to places where most people would not be allowed to. And so people like, yeah. Oh, yeah, I'm sorry. Yeah, that's right. You and Anna Maria and people from Whitehead, Rick and Rudy and Paul and many people have been there and met my sister, met my family. In fact, my father used to tell Anna Maria that, I'll be here next year. And so.
Has there ever been any pressure put on you to go back to Nepal to organize something educationally and higher education in Nepal?
Well, they invite me. and every so often I'd gone back. There's even a thing called Royal Nepal Academy of Science, believe it or not. But I go back and I get disappointed at how little gets done in spite of what you say or do or things like that. So I think that's, of course, they would want me to go back and see what I can do for them, but it doesn't happen. So I think I'm kind of resigned to the fact that I'll do my best here, and that that's. Angelica, right here. There's a seat right here. Sorry. Yeah.
Did you have any language issues? Was your college education?
Okay, yeah. So language issues. I grew up. I can't say I speak English very well, but I grew up speaking English. In college teaching was English.
Oh, so
In Nepal, yeah. In Nepal, at home, no. At home, I spoke the language, Nepalese language, because my mother doesn't speak English, for instance. So, but my father could speak English, and he, in fact, would speak in English to some visitors. But I had the person who taught me English he was also a teacher of English at the school where I went, and he was also related to me, and he had a son. He had a son who became a real buddy of mine at school. And so he eventually ended up being a physicist, went to England to do PhD and taught at the university in Nepal all the time. But so I used to really visit his house and get tutored in English a lot. So in terms of early education, grammar, and things like that, I think I got some guidance there. But that's really it. Otherwise, it's spending years in England and here.
Have your children maintained any of the Nepalese culture? They go back to visit the family?
Well, that's interesting question. I think the short answer to that is no. And the reason for it is that I am married to an English woman, and so when we came here, we could not go back to Nepal for many, many years. But my oldest son is very attached to Nepal. Yeah, he was 27 years old when I took him home, and he was incredibly struck at what he saw. And he was incredibly impressed by the fact that I could make it to MIT. from there. Yeah. And but he has been there on his own many, many times. And so I think he likes Nepal. He feels at home there with this big family there. The RajBhandary clan is about 300 plus strong. So we are all RajBhandharis because all the great grandfather, whatever, had 7 children, and so everybody is a RajBhandari. Everybody stayed until 20 years ago in the same compound. And so I think when Bob Weinberg visited a few years ago, he saw this area. And
I'm interested, Tom, about now. You had a nice secure position in Wisconsin, faculty position. What were the factors that led you to accept the offer to come to MIT?
Mm-hmm. Well, I've already said it. We can turn down MIT. Well, no, I think, actually we did think a lot about it because one is that the children were really happy there. And it's a lovely place to live. And secondly, Govind and I had worked together for so many years, and the fact that Govind was moving here and I was moving here, I consulted with many people as to whether this was a wise thing to do. There's some who said, no, you should not do that. Your name will forever be associated with Govind's name. You shouldn't do it. And others said, MIT is a great place. You'll establish your own program, and but Wisconsin did not make it easy. And I have not told you this. Wisconsin offered me tenure, when they knew that, I was likely to move. And so that did not make it easy. But I still, and I think after a lot of thought, decided that this was the best place to be.
When you made the decision to come here, Gobind had already decided to come here. Is that right?
Gobind had not decided, no. Gobind had not decided. In fact, I'll tell you now honestly that when I decided, Gobind was very happy. And then we celebrated that night, dinner, 4 of us, Esther and Joan and Gobind and I. Yeah, so this was, yeah, I think he had not decided to come.
Well, we were lucky.
Well, no, you're not lucky. I am lucky. I have enjoyed every minute of it. I think teaching has been a challenge, but interesting. It's always teaching is. I tell everybody, teaching is a learning experience, and every time I teach something, I learn. I think biochemistry. I have to tell you, when Gene took me to his office and told me that I would be teaching biochemistry with him, I had not taken a biochemistry course in my life. And yet, I had to teach biochemistry with Gene. At fairly short notice, I think. I don't think I got a year, Gene. I think it was pretty short notice, yeah. But, I think I looked at Vernon's notes, and I thought I could handle protein chemistry and protein structure and function, and nucleic acid chemistry and structure and function, and transcription, translation, replication. So, and it was, it was a lot of fun, Gene, teaching with you and learning how to teach. And Gene is very fussy. One of the things about Gene is Gene has his exams prepared at least 2 weeks beforehand. And I am a total procrastinator. I leave it till the last day, maybe, maybe the day before. And there was one time I almost did not make it. And that was, I was in England, and I was on my way back, and I had left my exam questions for the secretary to type. But I had not finalized it. And the exam was going to be the day after I arrived back. And this was the day that the Lockerbie accident, yeah, the plane that took off from London blew up over Scotland. And remember, my daughter absolutely petrified because she had heard the news that plane from London had been exploded, and I had just called from London to tell her that I was getting on the plane. I told her that because she had my car, and I needed the car to come to MIT after I arrived, so that I could look at the exams and finalize the exams. But fortunately, I was on the right plane, and got here, did the exams, and was very, very thankful. And I think I should learn from that I should never leave the exam till the last minute.
So, when looking back, maybe also for the younger people in the audience, when you look back, when you set up your lab, when you started out as independent investigator, junior faculty position, now sort of the other side of the fence, 30 years later.
Yeah.
Do you think it's harder now, or it's the same, or?
That's a difficult question to answer. Was it hard for me? I'll tell you what I did. When I arrived here, the first thing I did was to hire a technician. Yeah, I had been personally working in the lab myself a lot, so I think I knew, which is what the situation is with all of our junior colleagues here, that they're also, very much hands-on, have been hands-on. So the first thing I did was to hire a technician. Gene and Boris was the head and Gene was the associate head. They told me that there was one lab that had all kinds of equipment. This was a lab emptied by Bernie Gould, who's the other person? Cecil Hall, Bernie Gould, and one other person, Francis Smith. Yeah, everything had been put into one lab. So I had access to anything I wanted there. Old equipment, glassware, everything. So that's what I had. And I had a total of $75,000 spread over 3 years as my startup package. That's what I had.
At that time, that was a fair
Yeah,
amount.
yeah. See, he's very defensive. Yeah. Okay, see, I shouldn't say things like that. The provost, yes.
You were fortunate. I got no startup.
Yeah.
And I taught biochemistry from the moment I hit the ground.
Yeah, okay.
No warning.
Yeah. Okay, yeah. So maybe I shouldn't say a startup package because it was I was told that I would have access to some funds that the provost had. That's what it was. And the provost was Jerry Wiesner. Jerry Wiesner was the provost at that time. And so to go back to start. Yeah. So I hired a technician, and I think the first year I got 3 students, 2 from biology and 1 from chemistry. And trained the technician first, worked together with was able to work together with the students in spite of having to teach biochemistry. And once they got going, life became much easier. And it became a delight, in fact, to go into the lab. And Mehmet Simsek was my first student, and Joyce Heckman and Jim Ziegenmeyer from chemistry. Mehmet would be there smiling, and they were running all kinds of autorad. They were running 2-dimensional gels, 2-dimensional fingerprints, and there would be huge autorads, and he would be smiling with looking at the pattern and trying to tell me what the latest results was. And that is the joy of research to me. Every morning I would come to the lab, and there would be somebody with some result that they wanted to show me. And so it was a very, you work together with the students a lot. And actually, I did not have a postdoc until I got tenure, I think. I think I got tenure in 1973. Yeah. I didn't have a postdoc, so all with the students, and it was not a struggle for me. I enjoyed it immensely. Now, is it a struggle for our our young guys? I think they will probably have to answer that.
But there were no money worries. If you wanted a grant, you had a grant.
Oh, I did not have much grant at that time. I started with one NIH grant. Then I got 3-4 years after I got a Cancer Society grant, and grants were small. I think my first grant was $25,000 or something like that. Direct cost $25,000.
And you were paying for all that tuition for all those graduate students?
No, I don't believe I had to pay tuition for the graduate students at that time.
By 72,
69 is when I started. Yeah.
MIT was charging full tuition from, as soon as I could remember.
Oh, I think the tuitions, somehow I managed it. I don't know how, but somehow I managed it. But it may well be, and I remember, paying all kinds of exorbitant, what I thought was all kinds of exorbitant sums to MIT from the grant. But that's yeah. No, I think it was. We had to pay part of our salary, of course, from the grant, which is not half as bad as what some medical schools do, where you have to pay 100% of your salary from your grant.
So it was a breeze.
Well, I would not say it was not a breeze. I think I was very naive, Angelica, because I'll tell you some of the things that maybe I shouldn't say here. I never thought of tenure. To me, the challenge was just to do the best that you can, teach as well as you can, and just keep your lab going. And I was so just gung-ho on the lab and what was going on in the lab, and wanted to keep it going, that I did not worry about tenure. And in fact, the first I heard about my tenure was when I walked into the headquarters. It was a faculty meeting for one kind or another. And it's a very small room. People were sitting on the floor. I remember that. And Boris says, "Congratulations." I had no clue what the congratulations was about. And then it said that the academic council had approved my tenure. That's the first I heard about my tenure. So I was completely out of it. I was very maybe naive, or just too just concerned about just going to lab going and going to teaching going. And then I think that was a time when we had a visiting committee meeting. And at the visiting committee dinner that night, Rosenblith was the provost. Rosenblith told me that, congratulations, we just approved your tenure today. So that's when I heard about my tenure. That's how I heard about my tenure. Yeah, so yeah, I think I did not worry. Maybe I was naive, but things moved on. A research program got established, and I think I didn't stay with tRNA all the time. One thing leads to another, as you know, in research. That we got interested in mitochondrial biology for a while. Again, going from the tRNA, the lab developed. People in the lab really deserve a lot of credit for it. They developed techniques for sequencing tRNAs because the techniques that were available were just not good enough. Very sensitive techniques that led us to look at tRNA that ordinarily one could not look at, like mitochondrial tRNAs, chloroplast tRNAs, messenger RNAs, got us into messenger RNA analyses. The first mitochondrial tRNA was so surprising results, showed so many unusual features. We became interested in mitochondrial genes. That led to interest in nuclear mitochondrial coordinations, gene regulations, and things like that. And that lasted about 15 years, and without a grant on it. I was still using my main grant to support all that research. And I think we had at that time many students were interested in working on mitochondria. So I think that's how I kept that program going, through mostly through students, really. And the postdocs who came could bring their own money because that was an interesting project at the time. And then we became interested in, one thing I should say is to young faculty is that, sabbatical is one of the best things, that we have here. Is when 1975, after 6 years being here, I got sabbatical and went to England for a year. And that's really what changed my program, because I went there to learn suppressor genetics. And I really thought that helped my research program a lot. And that, in fact, where Bob was there at that time at the MRC labs, yeah. And Bob soon joined us. So sabbatical, unfortunately, I really have not stayed with taking sabbaticals because that was the first one whole year sabbatical, and the only other sabbatical I took was a trip to Japan where I took for 2 months. And that was the most enjoyable thing again, because I was working in the lab, it was working in the lab, and just, and I was just talking to Carolina, who is in the back here in the lab, that, one of the things faculty really miss is working in the lab, doing your own thing, but then we get too busy doing other things equally important. So who is to say, But I still envy the faculty who work in the lab. Mary Lou, Frank Solomon used to work in the lab. And I think I'm sure our young junior faculty who work in the lab. I'm sure they enjoy it a lot. Yeah.
I think, Tom, in the years I've known you, I think as much as anyone at MIT, you've been a little oasis of happiness and cheerfulness and at least relative calm. And I just wondered if you'd say a few words, because how you balance the rest of your life. I know family's been important to you, and most of you don't know he's an awesome squash player and stuff. I wondered if you'd say a few words how you balance that in the professional life.
I think, well, again, I have to go back to my father. One thing I learned from my father is that, never be angry. If you're angry at someone, try to think of why this person is behaving the way he or she is behaving. And that means that, it makes you think before you say anything in anger and things like that. And that gives you time to reflect. And that, I think, is incredibly useful to time to reflect. And if you're angry, not to say anything, but just think, why is this guy doing whatever he or she is doing? What it's doing. So that helps a lot, I think. And the other thing I have learned to do, and something I've felt essential to do, is that never take work home. That doesn't mean not to work at home. Never take, the problems that you have at work and all that, never let that affect your behavior at home. And vice versa. Never let the problems at home, which there are many always, bring that to the lab and affect your behavior in the lab. The two should stay completely separate. And that, I think, to me, helps a lot. Plus, the fact that I really have wonderful colleagues and wonderful students. And it's much better to be cheerful and have a happy face than, than otherwise. I think it's just, it's one of these things you learn. It's almost a survival instinct, if you like.
Did any of your children become scientists?
No. I think they saw how much of a commitment it required to be a scientist. I think. My oldest son is an architect. Not architect, I'm sorry. He went to Arts Institute, and so he is in California. And my daughter, she went to California, didn't like it. So when she finished school, came back, and she went into banking, and now she has a small business of her own. So, but they still follow, or try to follow what I do, or at least try to show an interest in what I do. Yeah. Yeah. So what else can I say? Is there any questions? Any other questions? I'm certainly happy to answer. But I think it's been life of fun. It's still wonderful to do science, and I still hope that I can continue to do science and continue to teach and be here in the department and enjoy being with my colleagues. And
you are currently associate head of the department. Do you like that?
Do I like it? Yeah. I think it's wonderful to be useful. Chris has been very kind to me, and so he has really shouldered most of the burden himself. But nevertheless, I think it's good to be useful and to feel that, I think one of the things I have this, in answer to Angelica's question, has life been just easy for me. I think I have always asked myself the question, do I belong here? That is a question that you ask when you come here. That's a question I still ask. Do I still belong here? Because I think the day you don't ask that question, you don't belong here anymore. Yeah. So I think, therefore, it's good to feel that you are doing something for the department, for the greater good of the institute. What's more important to me is doing something good for the department, and to serve in any way that one can, and to the best of one's ability, I think.
Right answer.
Right answer. Don't know about that, but I think once again I have to say that it's really been a joy. It's been a joy to teach so many smart students and to have so many people in the lab who have contributed so much and have kept my research program going, and to interact with so many colleagues who just are always helpful, supportive, and just work together well. Yeah.
Talk in a little more detail about the early tRNA work.
Early tRNA work. Okay. So, can I talk a little bit about the early tRNA work? Okay. So, one of the things that really thrilled me was to sequence one of the tRNAs by myself. Okay. That was the yeast phenylalanine tRNA. This is the one that everybody has done crystallography and all that stuff. And John Abelson, when he came here to give a seminar, said that he doesn't know how on earth I identified all those modified bases in those, that particular tRNA. We had developed some methods in Gobin's lab to tag ends of nucleic acids. And so that's really what allowed us to begin tagging, chemical tagging now. And then when polynucleotide kinase came, then we could tag it enzymatically. And so that is what allowed us to develop new techniques that could really be, in other words, instead of purifying grams of tRNAs, which we had to do in Madison, grams of tRNAs, and take 3 months to purify a tRNA, for instance. None of this affinity chromatography stuff these days. So that allowed us to really look at minute amounts of tRNAs, and we could just fragment it, label it, and then use the label to sequence it. In much the same way, same principles as Fred Sanger used to sequence insulin in the first place. Same general principles. So, but sequencing a tRNA was really, to me, the delight was, I was telling Carolina just the other day that we used to use chromatography to identify modified nucleotides. That's the only way you could do it, chromatography and spectroscopy. And one of the systems we used was a solvent system called isobutyric acid ammonia water. Now, those of you who are chemists know that isobutyric acid is just a horrendously awful smelling stuff. And who used it first? You might be surprised. Boris Magasanik in Shargap's lab, when they were quantitating the base contents of DNAs. So we used that, and I remember going to a Christmas party one night in Gobin's house, Christmas party, and my last bit of data that I needed to establish the sequence was in that chromatogram. Now, those of you who have not never used paper chromatography, this is a paper about this wide, about this tall, and it's drenched with isobutyric acid. Okay, so I get dressed. My wife is already at the party. I get dressed and go and take the chromatogram out. And I was not going to leave it to dry. I was going to look under the UV to see where the spots were, so I could identify what the spots were. Because by that time I knew what ran where, everything else. And so I went to the party. I knew the answer. I was really thrilled. I went to the party, and people would. So I remembered very, very vividly that, and feeling a little uncomfortable, but also really, really giddy about having the final data that I needed to. And that, I have to tell you, was a lot of work. And again, Jack Buchanan told me, we published 6 papers on that, back to back in JBC. And Jack Buchanan told me that when he saw those papers, he was convinced that they should hire me. So that was the early days, and then one thing led to another, and we started working with initiated tRNAs, and then that has so many unusual features that then we asked whether this was unique to all initiated tRNAs, and that led to collaboration with Mike Smith and all those guys, and then that led to work with mitochondrial tRNAs, chloroplast tRNAs, globin messenger. My first 2 postdocs worked with me on globin messages. That's another collaboration with David Baltimore. My postdoc collaborated with David Baltimore on the first cDNA synthesis, because globin message was the only purified message that was available then. And so therefore, Inder Verma in David's lab used reverse transcriptase to make this first cDNA ever. So that was a collaboration with David Baltimore, with my postdoc Gary Temple. So it's been a lot of nice collaborations, and people like Graham right down the hall, and just Mary Lou next door, Gobin, Bob 2 floors down, and so just wonderful. And I think this new building is a great building, and I think it was just. I have to tell you, you probably don't know that in the old building, I was not in the main biology building. I was in the chemistry building, building 18, but attached to biology through the bridge, the 5th floor bridge. And when I first moved to biology, my office was next door to the headquarters, 56503, which is the biology building. And my lab for the first year was in biology. Then when Gobind arrived and the building was finished, then I moved to the Gobind, to the whole floor. Gobind and I had the whole floor of building 18, fifth floor. So lots of memories.
Most of them good, I hope.
They're all very good. They're all very good. I think, life throws its curves, but I think you learn to go with it, and they've been very good. Yeah. Well, thank you very much for coming. This is really wonderful to see you all. Thank you.